Hi, my name's Chris Holder. I'm a bioinformatics workflow developer in the Epitome team. And today I'm going to be talking about some QC workflows we have in the biopharma production. So as you've heard in this session, we offer a number of different bioinformatics solutions, whether that be Dorado with expansion into secondary analysis that allows users to integrate these into their own pipelines, to the Epitome workflows that allow users, regardless of their bioinformatics experience, to analyze their data. And more recently, we've been working in applied markets, where we're creating workflows that allow actionable insights into key attributes of the data. And we offer these through our integrated end-to-end assays. Today, I'm going to be talking about two of these, the quality control space for biopharma production and how they can be used in mRNA-based therapeutics. So a little bit about mRNA-based therapeutics. They've been around in biopharma for a number of years in development. They can be split into two broad categories. The first one is a fixed antigen approach, like the COVID-19 vaccine, where every formulation has the same mixture of RNA molecules. However, they show huge promise in personalized therapeutics, where each formulation is unique to an individual for diseases such as cancer or rare genetics. So here we have an overview of what a typical personalized cancer vaccine workflow might look like. Starting at the top, which we call the discovery phase, where we start with an individual and we take a sample of the tumor and the blood. That is then sequenced on a high-throughput device like a PromethION, and then we analyze that data to identify neoantigens, and these are antigens that are present only in the tumor and not in the healthy cells. Once we select those neoantigens, they undergo immunogenicity screening, where we want to identify the neoantigens that elicit the highest immune response, that will have the best use in the formulation. And then in the bottom, we go to the regulatory phase, where the QC testing is based around, and that is around the mRNA-based formulation itself. So as you can imagine, with these personalized approaches, instead of making one large batch of the same formulation, you're making much smaller batches, but many more of them. They also want a typical turnaround time to be much shorter due to the nature of the diseases themselves, where we want the production cycle to be completed in a matter of months. So this really facilitates the need for fast, scalable production techniques, including the quality control testing. So anyone who is watching LACMOR talk today will be familiar with this slide. So this is looking at the quality control test of one of the for mRNA-based therapy. And it can be split into three broad categories, which is the sequence identity, the molecule integrity, and the purity of the sample itself. And each of these testing categories has a number of critical quality attributes associated with them. And these form part of the good manufacturing practice or GMP production that these labs adhere to. And basically, that's a set of standards that ensure that these products are made consistently, safely, and to regulatory standards. So if we were to do this with traditional methods, it can be quite laborious because for each attribute, it would require its own separate assay. And so that has a long turnaround time just due to the nature of the amount of sample prep, sample processing, and analysis that's required. This really becomes infeasible when you want to scale up to these personalized production approaches. So we really need to find a solution that is able to meet these demands. And this is where Oxford Nanopore fits in. Because of our unique ability to perform direct RNA sequencing, we realized we could create a test that covered each of these attributes and put it into a single assay. So that massively streamlines the testing process, the sample preparation and processing, and creating a system that's scalable to meet these production demands. Many of these attributes are already available in the current version of the workflow. However, some are still in development and will be released in future versions. So these assays exist on our Q-line gridiron, which has been purpose-built for applied and regulated environments, and the features that reflect this, such as being built to ISO 9001 standard, having enhanced support packages, and defined product life cycles. It's also been enabled to allow easy GMP compliance with features such as permission-based login and clear audit trail logging. The user interface of these devices is much simpler, where a user simply selects which assay they want to run, and it will integrate the sequencing, base calling, and analysis into one streamlined process, and that ensures consistency across analytical runs. So now I'm going to talk about the analysis part of each test, starting with the plasmid precursor. So plasmids form an important part of the biopharma production as they contain the DNA template of the mRNA we want to synthesize. And we need to make sure that the plasmid is of sufficient quality to ensure this effective synthesis. That includes features such as linearization, that the plasmid is properly prepared for effective transcription, the sequence identity, and that your sample is free from any contaminant DNA, whether that be from the host itself or other production sources. Like all our epitome workflows, this also produces a comprehensive QC report, such as this plasmid visualization map, so you can see that your plasmid has been constructed correctly and that all genetic elements that you expect are there, as well as a contamination metric. So if your sample does have contaminant DNA sources, we can find how much of them there are and the origin sources of them. So next, I'm going to be talking about the mRNA quality control test. And like I said previously, the heart of this test is our ability to perform direct RNA sequencing. This means we can sequence the full-length native RNA, including any modifications. With our latest kit and base caller, we are able to accurately estimate poly-A tail lengths, which is an important factor in these mRNA-based therapies as they control the stability of the molecule itself. We can also create consensus sequences with a per base modal Q-score above 45. And Bonja Boyden, who has worked extensively on the RNA base calling, is here on the panel if you have any questions relating to that later. So once we've synthesized our mRNA, we need to make sure that it is safe for therapeutic use, and that includes metrics such as the sequence identity, the integrity of the molecules themselves, and like I said, the poly-A tail length is expected. The workflow allows you to base call canonically or in the presence of N1-methylpseudouridine, which is a common modification used in mRNA-based therapeutics that increases the translation in vivo. The workflow can also be run in multivalent preparations, and so these are samples where there's a mixture of unique RNA molecules within them, and the workflow will separate these molecules out based on user-provided references and characterize them individually. Some of the comprehensive QC reports you'll get from this workflow are a histogram of the poly-A tail length distribution across all your RNA molecules within your sample, as well as a series of base composition plots. So at the bottom, we have a Sanger-style plot where you can see the nucleotide composition at each position, as well as, and in the middle, we have a per base Medaka consensus Q-score, and at the top, we have the coverage at each position. So at the end of both workflows, you'll get a PDF report. So the workflow will characterize your samples across a number of key attributes. A user is able to provide a threshold that is required for a sample to pass that particular attribute based on a user configuration file, and these can be based on their own internal validation and verification. We then integrate that user configuration file into the workflow, and then it helps produce this easy-to-interpret PDF report, which can simply state whether your sample has passed or failed all the attributes. So I hope this brief overview of these quality control workflows can show you the increased versatility of Oxford Nanopore sequencing. These projects have really allowed us to create a framework for us to create more of these end-to-end workflows. We are identifying unique current problems in applied markets and creating unique solutions for them, and that we are able to create actionable insights into our data. Thank you.